Rahim. Assalamu alaikum. Grade 7, subject biology, chapter 10, disease, page number 139. Student learning outcomes, bacteria, bacterial growth, and also today inshallah we discuss some bacterial shapes. So let's start today's lesson first. What is bacteria or define a bacteria? Bacteria are cells that are big enough to be seen under the light microscope, not by naked eye. Bacteria are found everywhere in the air, in water, and in the soil. You can say that they're present everywhere around us in all the three means, like in atmosphere, in soil, and also in water. They can also be found inside living organisms. Everywhere in the living organisms, they can found. Some bacteria are useful, but other can cause diseases, such as pneumonia, scarlet fever, and tuberculosis. These are few types of diseases that specially caused due to bacteria, or you can say that these are the bacterial diseases. Now let's study a little bit the structure of the bacteria and going to see inside what present of a bacteria. The basic structure of a bacterial cell has a cell wall, but not made out of cellulose as in plant cells. So you can see here the cell wall is present, but this not same as plants, because in plants the material is different from the bacterial cell wall. There is no proper nucleus, just a loop of DNA. You can easily see this. The loop of DNA is present here. That's not like a nucleus that present in animal or plant cells. Many of the structures found inside other cells, such as mitochondria, are missing in bacteria. So it means there is no energy or powerhouse in the bacterial cell. Bacteria often have additional loop of DNA inside their cytoplasm called plasmids. So you can easily see the plasmids that here, that just you can say that an extra loop of DNA. Bacterial growth. The next is how bacteria can grow. Bacteria can be grown in sterile condition in the laboratory. So page number 348 is mentioned. So I also choose a diagram for you. Let's see. A colony of bacteria start with just one cell. In the right condition, the cell divides to give two cells. Two cells become 4, 4 become 8, and 8 become 16, and so on. They're going to make a cluster of cells there. For example, you can see here this is the one cell. When it wants to grow, or a feasible condition in which it can easily grow, so one is divided into two cells. Two cells divide into 1, 2, 3, 4. These 4 cells divide into 1, 2, 3, 4, 5, 6, 7, 8 cells, and the 8 that divide into 16. So in this way, this growth is very rapid. Bacterial growth can multiply very quickly to form a colony. If you look at the graph, you can see 4 main phases in the growth of bacterial colony. The first one named as lag phase. So this is the lag phase where the growth just starts. Little growth occurs since the cells are taking up water and starting to make enzymes. So it's, you can say that just the initiation phase here, they just start and just gather the food, water, whatever they required, and make up their enzymes for the growth or make their size, or make their you can say that colony big. A log phase, when the population is increasing rapidly, the population increases by doubling and there is no shortage of food and water. This phase, log phase, here they have lot of, or you can say that enough amount of food and water, so they can grow easily, and they just double the number of themselves, okay, from like this 1 to 4 and 4 to 8 and 8 to 16, and you can see that here the slope is how going fastly. The next one is the stationary phase, when bacterial cells are dying at the same rate at which they are being produced. Here the number of bacterial cell, how many or how much produce, the equal amount of the bacterial cell going to die, or you can say that they are going to finish from that situation or in that condition. So we name as stationary phase. This may be because of shortage of food or because waste products are building up. As you know that as the growth is going on here, maybe the factor if no food there, so how the new generation and the upcoming bacteria what they can eat, so they become dying here at the stationary phase. The death phase, when more cells are dying than are being produced. As you know that the slope here going down, what happened here? The population decline cause of death may be lack of food, shortage of oxygen, or a build up of toxic waste products. So what happened after stationary phase might be possible no food here, and then the growth rate is not as similar as here. So what happened? They become going to dying and their amount and number of bacteria become decreased and they may be declined. So in this way all the growth, bacterial growth, you can understand this with the help of this graph here. It started from lag phase, then log, after that stationary, and the last is death phase. Bacteria which cause disease can survive harsh conditions such as heat, cold and drought. They can rest for a long time as spore. When good conditions return, they germinate and multiply rapidly again. They just wait for a favorable condition. As the condition reads, they again start to germinate, or you can say that they can again multiply themselves and rapidly they can increase in their numbers and form their colonies. There are three main shapes of bacteria. Bacillus rod shape, you can say this one. This condition is the rod shape in which bacteria you can see in this form also, in this shape also. Second, coccus or sphere, they are in the round form. This, the second shape, bacteria can also found in this shape. And spiralium or spiral, like this here, just like a helical, or you can say that in the form of a spiral. You can find or see the bacteria in these three forms mostly. So these are the main shapes of the bacteria: rod shape, round shape or spiral shape.